Hello, greetings. My name is Austin D. Clement, and my project is on botulinum toxin type A, also known as Botox. So here is a basically a graph of the various parts that makes up this mechanism and how this whole interaction is. So we're going to start off to the left, big picture. You have the myelin sheath surrounding the axon of the motor neuron, or motor neuron itself, should I say. But these are the myelin sheath. This is the axon terminal. These buds are the synaptic end bulb at the neuromuscular junction. This red arrow that looks like a lightning bolt, that is the nerve impulse that drives that action potential. This itself is the sacrament, and then these are what makes up basically the sacral the sarcoma, excuse me, and they basically facilitate in muscle contraction. So that's just something to keep in mind. And over at this little synaptic end bulb, when you go up closer, you see the nerve impulse again with the little arrow in red, driving the action potential. Then you have the synaptic vesicles containing the acetylcholine. Then you have the synaptic cleft, which is this gap here between the synaptic end bulb and the again sarcoma, and then you have in the pink the sarcoma, and then you have the receptors here. And when you go in further, and as you can see, those acetylcholine in red going out through exocytosis. And when you zoom in, you're gonna zoom right in to on to the right. The red or hot pink is the acetylcholine, and these are the vesicles that they are in. Then you have the synaptic vesicle, which is, as you can see, is heading towards the very end of the bulb. And then you see the exocytosis in these two bulbs, and the acetylcholine is leaving the bulbs or the vesicles through exocytosis again, and then binding. The acetylcholine is binding to the receptors, which opens the channels for that sodium concentration to come in. So, and which starts a reaction of muscle contraction in cell on neurons. That sodium will drive another action potential that was drove by the neuron. And so that is as it pertains to the synaptic bulb. But you might ask, what happens when there seems to be what happens in a state in which the cell is just pumping out this acetylcholine? What happens when that happens? Well, that is what one would call the pathogenic state, when things aren't going right, when there's a disease involved, and this disease is just constantly pumping out acetylcholine, which can cause a plethora of issues, whether that's eye twitching, excessive sweating, migraines, overactive bladder, muscle spasms. That's when that happens, because you will have this rush of acetylcholine. And if there's a rush of acetylcholine, all of these, as you can see, acetylcholine binds and a little sodium goes through. Well, all of the receptors are going to bind. So you're going to have this flush of sodium going straight through, causing these spasms, causing these reactions, causing these excessive sweating, overactive bladder, migraine, all sorts of things that goes on because it's constantly contracting, even when it's not meant to be so excitatory. That happens when there's again a rush of acetylcholine. And so, what is the treatment for this? What is this treatment for all this rush of acetylcholine resulting in all these different issues? Well, the drug Botox, as we know it, is supposed to alter the pathogenic state. So, as one can see to the top left corner, the neuromuscular junction with the axon terminal in blue, and in again pink is the muscle cell. The little hot pink inside of the green vesicles are the acetylcholine, and one could see that the acetylcholine normally just naturally leaving the cell through exocytosis. So let's look at it a bigger. So let's look at the whole left side of the diagram now, and it's titled normal transmitter release. So in a state of normal transmitter release, or even excited like over in a pathogenic state when acetylcholine is being flushed out a lot into the synaptic cleft. Well, we have again the synaptic vesicle with the acetylcholine in red, and that you can see this snare protein. And snare protein has three, or even also known as snare complex, has three separate but distinct different complexes onto it, or proteins onto it, and it binds to form the snare protein / snare complex. And one can see in purple Santaxin, and in yellow SNAP25, and in blue you have the synaptobrevin. And the synaptobrevin is in with the synaptic vesicle, and it's the protein that is a part of the cellular vesicle membrane. SNAP25 and Synaxin is already together, with Synaxin being a part of the actual neuron membrane. And so to form the complex, you need, and to even have exocytosis occur, you need the synaptobrevin to bind and form the snare protein or the snare complex. And when that happens, there's a chain reaction happen in which exocytosis occur, as you can see, and there's a release of acetylcholine, and then the acetylcholine is going to bind to the acetylcholine receptors, and these channels will open up by their binding, and they will go through the synaptic cleft and bind to that of the muscle cell acetylcholine receptors, which will cause the reaction and could cause a normal reaction, or what is considered to be normal, and release and have an excitatory response, but won't cause spasms. But it could be in a pathogenic state, and when there's a flush of acetylcholine, which would cause that pathogenic state. Now, how do you treat that? How do you alter that state? Well, you have the action of Botox. So what's going to happen is the Botox neurotoxin is going to bind to the receptor, to the botulinum neurotoxin receptor. And that will form endocytosis. So the other one was exocytosis, letting the acetylcholine to go out. But this one is going to form the endocytosis, which the vesicle is being formed. And it's going to be formed to allow that Botox or botulinum neurotoxin to go inside the neuron. And then, as you can see, and it's once inside, that heavy chain That heavy chain is going to be a heavy chain, light chain dynamic. So that heavy chain is going to go help facilitate and going to facilitate that vesicle creation. And then the light chain will go outside of the vesicle, and then boom, it cleaves the snare proteins. It cleaves syntaxin. It cleaves SNAP25. And it prevents snaptobrevin from even binding because that's cleaved too. So once snaptobrevin is cleaved, then all you have is acetylcholine in these vesicles. And if once these complexes are cleaved and they cannot bind, so that means exocytosis of the acetylcholine cannot happen. So acetylcholine is not going to go into the synaptic cleft, and as a result, it's not going to bind to the acetylcholine receptors on the muscle cell. So all those issues that was going to occur will no longer occur because you have acetylcholine blocked. Not acetylcholine blocked, but you will have nowhere for acetylcholine to bind, or should I say, synaptobrevin to bind to form the snare protein or the snare complex, which means that exocytosis of acetylcholine will not happen. So that's the basic pathway. And this is a basic map of this pathway. So what type of experiments can be done? So to understand, can actually, and the question is, and the question that was proposed, was how can one prove where in the cell the drug acts? How can that happen? Well, you have the snare proteins, and the snare proteins is in the axon terminal, or should I say, I'm sorry, excuse me on that, is in the neuromuscular junction, specifically in the bulb, in the synaptic bulb. And so how do you prove that? Well, these are the three proposed plans that I was debating as a possible experimental plan. You have ELISA or Western blotting in cell lysase to determine if snare proteins were cleaved. That's one. Then you have 2, toxin activity determined by cleavage specific antibodies in live cells with the facilitation of immune fluorescent methods. That's 2. And then you have assessment of neurotransmitters being released in neuronal cell cultures. That's 3. So the one I chose was the use of ELISA. And I'm about to explain why I chose that and the steps, of course, of how to do that for our experimental plan. But one might ask me, why didn't I do the other 2? Well, actually, there was multiple proposed plans, but those plans were for test numerous things, but these are the 3 specific ones that I was thinking of. Because yes, one could try to do a variety of things like measure calcium intake to figure out, does it bind to calcium? But we already know. The question is, can you prove it? So I could look at acetylcholine receptors being inhibited, but this is how you can prove specifically that it's in this place, in this direct place. So it's under my understanding that this plan has to be about what I know and what I believe and what I think, and that is the snare protein complex, like many researchers have already suggested. So for that reason, I thought those other proposed ideas, of course, won't happen. So the toxin activity determined by cleavage-specific antibodies in live cells with the facilitation of immune fluorescent methods. I've looked several research articles on that, and they use that method to understand those dynamics. However, they had troubles with this method. Although it was like they were answering the questions indirectly. They did not specify why they answered those questions indirectly, but they answered those questions indirectly and not being as concrete. And And one could say, why couldn't you do, since it's somewhat of a ELISA type Western blotting, couldn't you do a mechanism in which it's more direct? But the research didn't say that, so I decided to take that off the table, even though I know that could happen. But in my mind, there must be a reason why the researchers didn't have that approach. Like, what went wrong? Did something? Why not do that? Maybe there is a knowledge gap that I'm not familiar with. So that's why I left that alone. Assessment of neurotransmitters being released in neuronal cell cultures that have been seen using thawed neurons and experimenting on that and dissections and things like that. But the thing was, it's the goal. The goal is to find a plan that specifically identifies snare protein. If that's what we're sticking with, we're gonna have to stick with the snare protein, and that does not answer with the neuronal cell cultures. That does not necessarily answer the question or prove the exact location. It does not, because the location, as I explained, could be anywhere in which neurotransmitter is not being released, or there's an inhibition going on in that synaptic area, in that synaptic bulb, dealing with, as I previously said, acetylcholine receptors being inhibited, or calcium intake being blocked by variety of different mechanisms. So that's why I chose ELISA. So ELISA testing, let's just get right into it. You have 4 major steps. Plate coating, 1. 2. Plate blocking, 3. Antibody incubation, and 4. Detection. Due to the time constraint, I'm just going to briefly talk on to the main points. The main points of how this idea would go about. So, ELISA testing, also known as enzyme-linked immunosorbent assay, has a basically what one is testing involves an enzyme or a protein that catalyzes a biochemical reaction. In this case, that enzyme or enzymatic causing protein, that would be the Botox. And then it also involves an antibody or antigen or immunological molecule that may form an antibody or antigen or antigen antibody reaction to provide a positive, or if no result at all, a negative result. This has been used for a variety of different diagnosing, like HIV, allergies, things like that. But in the case of the antigen antibody reaction, that's going to be the snare protein complex, or even those, as you can see, the snare the complexes that make up the snare protein or snare protein complex, the SNAP25, the syntaxin, the synaptobrevin. That's going to be that antibody or antigen or antibody antigen complex. And so the goal is, if it's positive, if the test is positive, then that means that that the synaptobrevin, SNAP25, and syntaxin has been cleaved. If no reaction occurred, that's a negative result, which means that there was no cleaving involved. So Botox did not cleave these things. So my proposal is, there's a 96 wells, 96 well per individual complex or individual protein. That's like SNAP25, synaptobrevin, and then another one is going to be using a control. Basically, that's going to be like a saline solution, and have that be that. And so, or even that's idea 1. Idea 2, you're going to have the snare complex and then just have the the snare protein complex and just have the Botox just cleave off of the complex itself. So those are what I'm proposing, and seeing that, and it's in 96 wells, so that should be enough. 96 well per control per uh, investigation of the other ones either being the snare complex itself or the individual bodies that make up the snare protein complex. So that should be well enough. And the reason why I that one would need a lot is what is going to be discussed shortly. And that's why I chose ELISA testing, because out of all the research articles, out of all the techniques, ELISA testing, when it comes down to snare complexes, understanding snare protein complex, and when it comes down to Botox, the protein, it seems like ELISA testing was the most useful. Although this is my opinion, the research also would back me up and would back that opinion up as well. So, but I still had to make sure. I still had to really understand, and this is a direct, just to specify, direct ELISA test, just to specify that, and also the indication on positive and negative with the enzyme immunoassay would be the indication use of color, could be, or it could be something along the lines of actual seeing cleavage by doing, if you did a hybrid like a staining and being able to see that as staining on a plate and using dye for color to change the enzyme being labeled for the antibody in this case and being able to see that. And I just want to add that just to make sure I cover everything. And then so as it relates to the benefits that I had to think, like why would I choose ELISA testing over the other ones, I have to look at the advantages. It's a fairly simple procedure, so it's easy to perform. It has a high specificity and a sensitivity, and that's because it's based off of antigen antibody reaction. It's high efficient, so because the analysis can be performed without any complicated sample pre-treatment. It's when you look at being cost effective in terms of reagents, it is relatively on the low cost, again in terms of the reagents. And of course, we always have to make sure that this is a generally safe and eco-friendly procedure, which it is, because no radioactive substances and large amounts of organic solvents are used, or are required, should I say. However, the disadvantage of ELISA testing with all the other tests that I have looked at was that it is labor intensive due to the timing. You have to get timing right. Pipetting, of course. The incubation process and the different incubation times. And that's why I did not put that up here, because you have to really look at it specifically and look at the incubation specifically and look at the buffer system, which you can use different uh, different ways in which you can construct it. But this is just general understanding of it and why in which I would use this, and exactly, even you could use even a PCR test if you are so inclined. But but back to ELISA, uh, it is although it is easy to perform and it's a simple procedure, it can be considered by some a little on the sophisticated side. And when it comes down to expensive, although reagents are relatively low cost, the culture media is expensive and is required. When it comes down to high possibility of false positive and negative, it does come into play. That comes into play, and that's one of the reasons why I was so careful with thinking about this and the use of ELISA testing, because that could be a potential issue. But with all, that's why the 96 wells come into play with this, because that's when you get a larger sample of data, the more that could be extracted and the more definitive your answer can be. With Elijah's test as it relates to this and the Botox and Botox research, I did notice in the studies that there could have been, and as it relates to the SNAP complex as well, there is considered to be antibody instability or insufficient blocking of immobilized antigen results in false results. So that is something that has to be watched out for, very careful. So all the procedures must be very thought through and very carefully planned, and also an understanding of exactly what Botox, what this procedure need when you consider Botox and SNARE complex, the SNARE protein complex. And then yeah, so that's one of the big things that I looked at the research and I thought needed to be recognized. As it pertains to another disadvantage, which isn't so much of a disadvantage, was it requires refrigeration and refrigerated transport and storage. But however, that I don't see as a larger problem as the other plethora of, not too much plethora, but enough that you have to be very careful. So that's my test, and that's my plan, and that's how I thought it out. And I hope that really brought a lot of insight into how I was thinking and going about in planning an experimental process of this. As it relates to how Botox modulates and the mechanism by which it works, I hope I already somewhat explained that. But as it relates to, as I said, wrinkles, eye twitching, migraine, muscle contractions, excessive sweating, all of these, and even what's not up here, the gallbladder, as it relates to all those unique and interesting, unique and interesting situations, one must understand that Botox is considered to have high toxicity. But in low dosage, it has the ability to treat various clinical conditions, as said all the conditions before. And type A and type B are the most commonly used Botox for medical purposes. So, as it relates to, I'm going to start with excessive sweating. As it relates to excessive sweating, acetylcholine facilitates the sympathetic neurotransmission in the sweat glands. And because of that, injection of Botox could inhibit the excessive sweating by overall preventing the release of acetylcholine. That means if acetylcholine is not being let out of the synaptic cleft, sweating will probably not occur. It won't occur. It will prevent it from occurring. As it pertains to muscle contractions, Botox can treat multiple overactive muscle disorders. These muscle disorders include post-stroke spasticity, spinal cord injury associated with spasticity, head and neck spasms, clenching of muscles of the esophagus, jaw, urinary bladder, and other type organs. And as you can see in this diagram above, like I showed you before, if acetylcholine isn't going in and sodium, if acetylcholine is not leaving by exocytosis and not binding to the acetylcholine receptor, opening up the sodium channel, then that means muscle contraction will not happen. The muscle contraction, again, will not happen if acetylcholine does not bind to the channel. And if that doesn't happen, then a muscle contraction won't happen. As it relates to migraines, if you inject low dosage of purified Botox around the pain fibers, then the release of chemicals like those facilitated in pain transmission, like acetylcholine, will not be released, which will reduce the incidence of migraine and which will prevent a migraine. And also, if acetylcholine not being produced or chemicals like that not being produced, then incidence of migraines will be reduced. Eye twitching relates to muscle contractions. But let me just specify with eye twitching. When you have eye twitching, when anyone has eye twitching, and there's a muscle contractions, and acetylcholine facilitates that. If acetylcholine cannot, if those vesicles with acetylcholine cannot perform exocytosis, and acetylcholine is in the synaptic cleft, then eye twitching won't happen because muscles need to have the gates need to open. And if the key is acetylcholine and the door is those sodium channels, if you don't have the key, then you cannot open the door. And if you cannot open the door, then nothing's going to happen. It's just going to stay a door. Nothing's going to happen. It will stay in that state. Now, I'm going to save wrinkles for last, and I'm going to go on something that's not up here, but overactive bladder. People or patients with an overactive bladder could be treated with Botox. And they could be treated with Botox because Botox can be injected into the walls of the urinary bladder. This will reduce the urge for frequent urination. When Botox is introduced into the urinary bladder, Botox prevents the vascular release of acetylcholine, reducing the urinary bladder contraction, which in turn will benefit the patient because there's a refractory in the overactive bladder. And for this reason, and for all the reasons above, whether overactive bladder, migraine, excessive sweating, muscle spasms, eye twitching, Botox is all approved for all of these by the FDA, Food and Drug Administration of the United States, as an approved drug for treating these ailments. Now on to one of the most well-known, wrinkles. Facial wrinkles has been used as and is considered to be a safe mechanism for reducing facial wrinkles. It has been shown to relax wrinkled muscles, which in turn results in a smooth overlay of skin, and is considered providing superior results, being absorbed after just a few repeated injections. Botox selectively binds to the peripheral cholinergic motor neuron end plates to prevent the release of acetylcholine. And just to let you know, as mentioned, the motor end plates are here onto the right of the image, and once it binds to the receptors, and these are the end plates, so it's basically, as I said before, it binds to it, which prevents the release of acetylcholine. However, Botox has been known to paralyze involved muscles for a short period of time. Research says up to 3 months. However, the restoration of muscle function can be absorbed shortly after the gradual formation of new motor end plates. So excuse me, that's how it works. And this is the last graph of it, and I hope I showed it to you, and I hope I answered your questions. I hope I gave, as you can see, the mechanism of action, briefly summarized what the normal cell biology, explain the pathogenic state, explain how the drug alters, drug meaning Botox, alters the pathogenic state. Hopefully, I explained with the use of a figure depicting a pathway map and indicating the point at which the drug acts, and also developing and presenting an experimental plan by which one can prove where in the cell the drug acts. I hope I explained that as it pertains to the ELISA testing, why I chose it, how the ELISA testing works, things of that nature, and all the experimental plans that I've thought, and explaining why I chose ELISA specifically and what ELISA does. As it relates to depicting what cellular functions it modulates, I hope I explained that as it pertains to the wrinkles, eye twitching, migraine, muscle contractions, excessive sweating, gallbladder. I hope I explained that well, and the mechanism by which it works. I hope I explained that well as well. And this is the last graph, just reminding you of, brief overview of how it works on the cellular level. This is my references. These are my references. I hope that you enjoyed this presentation. I know it went a little bit over time, but that's my explanation and that's my presentation. This class has been great. I learned a lot. Thank you again for all your work and all your assistance and everything. Thank you again.